Assalamu alaikum. Let's talk about the anti-mycobacterial drugs and their mechanism of action. We know that mycobacteria is a highly resistant bacteria because its cell wall is different from the other bacteria, and its cell wall is composed of four layers. The first layer is called peptidoglycan layer. The second one layer is called arabinogalactan layer, and the third one, the most important layer, is called mycolic acid, and the last one layer is called glycolipid, which is the outer covering of bacteria. So, due to its different cell wall, the mycobacteria is a highly resistant bacteria, and we need to combine various type of antibiotics to kill the mycobacteria. So, let's talk about the mechanism of action of various antimycobacterial drugs. The first one is the rifampicin. Inside the bacteria, there is a DNA, and the DNA is converted into messenger RNA with the help of an enzyme called RNA polymerase enzyme. This is the enzyme called RNA polymerase enzyme, and this enzyme produce messenger RNA from the DNA. And then the messenger RNA is used to synthesize protein with the help of ribosomes. But rifampicin block the RNA polymerase enzyme, and when the RNA polymerase enzyme is blocked, so no messenger RNA will synthesize, and thus no protein synthesis will be occur. Okay. So this is the mechanism of action of rifampicin. The second one is streptomycin. Actually, streptomycin block the small subunit of ribosome, which is called 30S unit, and by blocking the 30S subunit, streptomycin prevent the protein synthesis. Thus, protein synthesis will be stopped and bacteria will die. The third one is called pyrazinamide. So pyrazinamide work by blocking the enzyme called fatty acid synthetase enzyme. Actually, this enzyme is involved in the synthesis of a special type of fatty acid, which is then used in the synthesis of mycolic acid. This is a mycolic acid layer, and fatty acid synthetase enzyme is involved in the synthesis of mycolic acid. So pyrazinamide block the fatty acid synthetase enzyme, and thus no mycolic acid will be synthesized. This is the fourth one called depsone. Inside the bacteria cell, we have various compounds called para-aminobenzoic acid, and these compounds converted into dihydrofolate, and then the dihydrofolate is converted into tetrahydrofolate, and tetrahydrofolate is involved in DNA synthesis. So depsone actually block the pathway of para-aminobenzoic acid conversion into a tetrahydrofolate. So when tetrahydrofolate will not form, so no DNA will form. The fifth one is the isoniazid, which is the most important and first line drug against the mycobacteria. When the isoniazid enter into the bacteria cell, so it converted into active form with the help of an enzyme called CATG. This is also called the catalase peroxidase enzyme. This peroxidase enzyme convert the isoniazid into active form, and with the active form there is the combination of another molecule called NAD. NAD combined with the active form of isoniazid, and this combination then block an enzyme called anoyl reductase enzyme. So when anoyl reductase enzyme is blocked, so no mycolic acid synthesis will be occur, and thus cell wall synthesis will be blocked and bacteria will die. Isoniazid when enter into the bacteria cell, so it converted into active form with the help of an enzyme called CATG, catalase peroxidase enzyme, and with the active form there is the combination of another molecule called NAD. So this combination then block an enzyme called anoyl reductase enzyme, and this enzyme is involved in the synthesis of mycolic acid. So if this enzyme is blocked, no mycolic acid synthesis will be occur and bacteria will die. Sixth one is the ethambutol. Actually, it block an enzyme called arabinosyl transferase, and this enzyme produce arabinogalactan layer of the cell wall from the two compounds called galactose and arabinose. This enzyme combine the galactose and arabinose and produce arabinogalactan layer of the cell wall. So ethambutol block the arabinosyl transferase, and thus no arabinogalactan layer formation will occur, and thus cell wall synthesis will be stopped and bacteria will die. These are the first line anti-TB drugs. The first one is rifampicin, isoniazid, ethambutol, and pyrazinamide, and the brand name of these combination is called Myrine P. This was all about the antimycobacterial mechanism of action.